Welcome to the Deep Dive, where we take the latest research and distill it into the most important insights, just for you. Today, we're plunging into a problem that, well, it's often unseen, but it's silently doing significant damage to our planet's most vital resource, water. We're talking about nitrogen pollution, specifically ammonia nitrogen, or NHN.
It's truly a pervasive issue. This form of nitrogen pollution, it streams into our water from so many places, everything from agricultural runoff, heavy with fertilizers, to industrial wastewater from all sorts of manufacturing processes, and even just urban runoff from our cities.
And the effects are, well, frankly alarming. You might not realize it, but even tiny concentrations, like as low as 3 milligrams per liter, can be toxic to fish.
Yeah, it's surprisingly low.
Think about that. Just a tiny amount could start causing serious problems. And beyond that immediate toxicity, excess nitrogen fuels eutrophication, which leads to those explosive algal blooms that can basically suffocate entire aquatic ecosystems, creating these vast dead zones where life really struggles. The urgent need for effective solutions is, well, it couldn't be clearer.
Exactly. And for years, we've relied on traditional methods like biological processes, ion exchange, or maybe chemical precipitation to get this ammonia nitrogen out.
Okay.
But these methods, they often come with significant drawbacks. They can be complex to run, expensive, or sometimes they even contribute to secondary pollution by creating new waste streams.
So solving one problem creates another.
Sort of, yeah. Which is why researchers have been working really hard to find cleaner, simpler, and more cost-effective alternatives.
So given those limitations, what promising new avenues are researchers exploring? What works better?
That's a crucial question. And this is where adsorption really starts to shine.
Adsorption.
Yeah, it's a process where pollutants basically stick to a surface. It's attractive because it's simpler, generally cheaper, and doesn't usually create that secondary pollution problem. And the star material emerging in this whole story is biochar.
Biochar. Okay, that's essentially a special kind of charcoal made from biomass, right? Our sources highlight it, and it sounds pretty cool.
It is, yeah.
It's not just good at cleaning water, but then after it's soaked up those pollutants, it can be repurposed as soil fertilizer, returning nutrients to the land. That's real resource recycling.
Exactly.
Sounds almost too perfect. So what's the catch? What's the big challenge researchers have faced with it?
Well, it is incredibly promising. If you connect this to the bigger picture, biochar has these great properties, like a really large surface area, a well-developed pore structure. It acts like a super effective sponge. It uses both physical mechanisms, like electrostatic attraction and ion exchange, and chemical ones like surface complexation to grab that ammonia nitrogen. But the bottleneck, the real challenge, has always been optimizing its performance.
How so?
Well, imagine trying to test every single combination, different types of biochar, different water conditions, temperatures, pH levels. Doing all that with traditional lab experiments, batch experiments, is incredibly labor-intensive, time-consuming, and honestly, pretty costly. It makes it really tough to figure out the complex interplay of all the factors influencing how well it actually adsorbs.
Right. Too many variables, not enough time or money.
Precisely.
And that is our Deep Dive's mission today. We're exploring how cutting-edge machine learning, or ML, is being used to overcome these exact challenges. Think of it like giving biochar an intelligent brain, basically allowing us to predict and optimize its effectiveness in removing ammonia nitrogen from water, but with unprecedented speed and accuracy.
Okay, let's unpack how combining, nature's solution with this intelligent tech is giving us a shortcut to cleaner water. So we've established the urgency, right?
Right.
Nitrogen pollution is a big deal, and biochar offers this promising kind of eco-friendly solution for adsorption.
Yeah, like a natural filter that keeps on giving.
Exactly. But that complexity, knowing how to use it best under different conditions, that's been the real hurdle. Indeed. The challenge, as we said, was efficiently untangling all those influencing factors, the biochar's own makeup, the water conditions, temperature, pH, and so on, without just doing endless, costly lab work.
Right.
That's the research gap that machine learning is now helping to bridge.
Okay, so here's where it gets really interesting. The game-changing part. Our sources dive into how machine learning is transforming this field. Think of ML as this intelligent pattern detector. It learns from massive amounts of data to make predictions and, crucially, optimize outcomes. It's not just about filtering water. It's about intelligently designing the perfect filter for almost any given scenario.
And ML isn't entirely new in water treatment. Previous studies have used it for predicting contaminant removal, including ammonia nitrogen, but often with other adsorbents.
Like what?
Oh, things like zeolites or even waste foundry sand.
Yeah.
They've achieved pretty high accuracy with models like artificial neural networks, ANNs, for example.
Okay, so earlier studies used ML for similar problems. What makes this specific research on biochar so significant? Why is it different?
That's an excellent point.
Hmm.
What makes this study particularly significant is its laser focus specifically on biochar's adsorption performance for ammonia nitrogen. That was a previously limited area.
Oh, okay.
It directly tackles the heterogeneity of biochar, the fact that not all biochar is the same, and the complex interactions involved. And it does this by using a really comprehensive set of features,
Features meaning?
meaning the input data for the model. So it includes both the biochar's properties and the experimental conditions under which the adsorption happened.
Right, getting the full picture.
Exactly.
Hmm.
And crucially, they pulled together a significantly larger data set than most prior work. We're talking a whopping 417 sets of adsorption data from 46 distinct biochar types.
Wow.
Yeah, collected from a decade of literature, 2014 to 2024, across major databases like Web of Science, Google Scholar, Scopus.
That is a massive amount of data to draw from. So with this really robust data set, what were the main goals? What did the researchers set out to achieve?
They had 4 clear objectives. First, to meticulously process all this published data, get it into shape. Second, to compare a bunch of different machine learning models and pick the best one for predicting biochar adsorption.
Okay.
Third, and this is really important, to deeply understand which variables, which factors most influence that adsorption capacity, the why.
The interpretation part.
Exactly. And finally, to develop a user-friendly application, basically software, to put this intelligence directly into practice, make it useful.
Okay, building an ML model, it's like teaching a computer, right? You need good data, and you need to prepare it really carefully. This study definitely collected a massive amount of info. Can you walk us through the types of data they gathered?
Certainly. The data parameters, the features we talked about, were divided into 3 main categories. First, the chemical properties of the biomass itself, the stuff used to make the biochar.
Like wood chips or crop residues.
Yeah, exactly. So things like its carbon content, molar ratios like oxygen to carbon or hydrogen to carbon, and how much ash it contains. Second, the physical properties of the biochar that resulted its specific surface area, basically how much sticky surface it has, its total pore volume, and its own pH level.
Got it.
And third, the experimental conditions under which the adsorption tests were run. Things like the temperature, the pH of the water solution, and a key factor, the ratio of the initial ammonia nitrogen concentration to how much biochar they used.
Right, the dosage. Okay, so they had all this raw data from hundreds of studies. Now, before any AI can learn effectively, you have to feed it clean, well-organized data. What did the researchers do to prep this massive data set for the ML models?
That's absolutely critical. Cleaning up the data is just as important as collecting it. Think of it like a chef meticulously prepping ingredients before cooking.
Good analogy.
They focused on 4 key things. First, missing values. Sometimes a study just didn't report a certain value. Instead of throwing out good data or just guessing, they used a clever statistical method, KNN imputation, to intelligently fill those gaps based on similar data points.
Okay, so smarter filling in?
Exactly. Next, they applied a statistical technique called Box-Cox transformation to smooth out the data. Some data can be really skewed, and this technique helps make it more normally distributed, more uniform. That gives the ML models a clearer landscape to learn from, reduce skewness significantly for some variables.
Makes sense. What else?
They also make sure to remove any redundant information. Sometimes you have 2 variables that measure almost the exact same thing. That's called collinearity.
Ah, yeah.
Like, they found the O plus NC ratio was highly correlated with the OC ratio and had less impact anyway, so they removed it. This prevents the model from getting confused or sort of overemphasizing one aspect.
Streamlining the inputs.
Precisely. And finally, they scaled all the features. This puts everything on a comparable playing field using z-score normalization. It's vital because if one factor is measured in thousands and another in single digits, the model might incorrectly think the bigger number is more important just because it's bigger.
Right, apples and oranges.
Exactly. Scaling ensures a fair comparison and lets the model learn robustly.
Okay, so after gathering and meticulously preparing all this data, the researchers put 12 different machine learning models to the test, like a scientific Olympics, you said, to see which one performs best at predicting this ammonia nitrogen adsorption. Which types of models were in the running?
It was quite a lineup. The study systematically compared 12 models spanning 3 main families. There were 6 tree-based ensemble models, think random forest, gradient boosting decision trees, XGBoost, Light GBM, CatBoost, extremely randomized trees. These are like having teams of decision-making experts working together.
Okay, ensemble methods.
Then 3 kernel-based models support vector machines, kernel ridge regression, Gaussian process regression. These are often good at finding complex patterns in data.
Right.
And finally, 3 deep learning models, multilayer perceptron, convolutional neural networks, CNNs, and long short-term memory, LSTMs. These are the heavyweights, often known for handling truly massive or unstructured data.
So a real cross-section of ML approaches. How did they figure out the winner? What was the evaluation process like?
It was pretty rigorous. They split the data, 80% for training the models, 20% held back for testing how well they generalized.
Standard practice.
Yeah. And to fine-tune these models, to get their internal settings just right, they used a smart, iterative process called Bayesian optimization combined with 5-fold cross-validation.
Okay, what does that mean in simpler terms?
Think of it like a really efficient series of test runs. The model trains on a part of the data, gets tested on another part it hasn't seen, adjusts its settings based on the errors, and repeats this process multiple times in different ways. It helps find the best settings without the model just memorizing the training data.
So it learns general patterns, not specific example.
Exactly. And they even assessed model stability by running this whole process 50 times with different random splits of the data just to make sure the results weren't a fluke. Performance was measured mainly using RMSE root mean square error, which tells you the average prediction error.
Lower is better.
Right. And the R-squared value, or coefficient of determination, which tells you how much of the variation in the actual adsorption capacity the model could explain.
Closer to 1 is better there.
You got it.
So after all that rigorous testing, the big reveal, which model came out on top? Which one was most effective for predicting Biochar's performance?
Well, the results were pretty clear. The tree-based ensemble models consistently did better than the others. And the absolute top performer was CatBoost.
CatBoost.
Yeah. And this isn't just, a statistical win. It's potentially a game changer for the field. CatBoost achieved an impressive R-squared value of 0.9329 on the test set.
Wow, over 93%.
Exactly. Imagine knowing, with over 93% accuracy, how effective a particular biochar setup will be before you even run a single expensive lab experiment. That's a massive leap in efficiency.
That is incredibly powerful. So why CatBoost? What makes it so good at this particular task compared to the others, even other ensemble methods?
CatBoost has several key advantages built in. It has superior ways of handling missing values, and especially categorical features, things that aren't just numbers, which are really common in messy real-world environmental data sets.
Okay.
It also uses a symmetric way of building its decision trees and has these clever gradient-based balancing mechanisms during training. All this helps prevent overfitting.
Overfitting meaning it just memorizes the training data.
Exactly. It learns the specific noise in the training data instead of the underlying pattern. CatBoost is designed to resist that, ensuring it performs well on new, unseen data, which is what you really care about.
Okay, that makes sense. And what about the other models? You mentioned deep learning models like LSTM and CNN didn't do so well. That's surprising, given all the hype around deep learning.
It is a bit surprising at first glance, but it turns out those incredibly powerful deep learning models, they're often like using a sledgehammer to crack a nut for certain problems. They typically excel with much larger, often unstructured data sets. Think images or massive text data sets where their ability to map incredibly complex nonlinear relationships can be fully leveraged. Here, the data set, while large for biochar studies, probably wasn't quite massive or complex enough in that specific way for them to really shine. Their power was almost overkill, or maybe just not suited to this type of structured data.
So the tool needs to fit the job.
Precisely. The kernel-based models, they were stable, reliable, but they just couldn't quite capture the really intricate nonlinear relationships as effectively as the top ensemble methods like CatBoost did in this case.
Okay, so it sounds like CatBoost really hit the sweet spot for this problem. Was its reliability confirmed in other ways?
Absolutely. They did further checks. Stability analyses, looking at how performance varied across those 50 runs, confirmed its consistency. And residual analyses, basically looking at the errors, the difference between predicted and actual values showed the errors were small, clustered tightly around 0, and normally distributed.
Which is what you want to see.
Exactly. It confirms minimal bias and high accuracy, giving real confidence in the model's predictions.
Okay, so once you have the best model, CatBoost in this case, The next crucial step, as you mentioned, is figuring out why it's making those predictions.
Right.
What factors truly drive biochar's ability to absorb ammonia nitrogen? This is where the aha moments happen.
Precisely. Understanding the why is key. This study used advanced techniques, embedded feature importance, EFI, and SHAP values, which are like detective tools that quantify the impact of each input feature on the model's predictions.
Okay.
The results provided a really staggering insight. When they grouped the factors, experimental conditions contributed most significantly to ammonia nitrogen adsorption,
How much?
accounting for 67.2% of the influence.
Wow, really? Over 2/3?
Yeah. This fundamentally shifts the focus. While the biochar's own makeup definitely matters, its physical and chemical properties accounted for the other third or so.
Okay.
It's overwhelmingly how we use it, the surrounding conditions like concentration, temperature, pH, that dictates its success in absorbing ammonia nitrogen. That tells us where to put our energy for maximum impact when designing applications.
That is a powerful insight. So drilling down then, if experimental conditions are so dominant overall, what were the top individual factors the model highlighted, whether from the experiment or the biochar itself?
Looking at the individual factors, the analyses consistently pointed to 4 key players having the biggest impact. Top of the list was that initial concentration of ammonia nitrogen relative to the biochar dosage, C0.
Okay.
Then came the experimental temperature, temp, the carbon content of the biochar C, and its total pore volume V. These 4 really stood out.
Interesting. So how do these factors actually influence absorption? This study used partial dependence plots, or PDPs, right? And these are really pull back the curtain on the specifics.
We do, yeah. PDPs are great because they show how the predicted outcome changes as you vary just one feature while holding others constant. So for that initial concentration, C, absorption capacity generally increases as C-shirt goes up, but only to a certain point around 50 milligrams per gram in this study.
Why is that?
Well, initially, a higher concentration creates a stronger driving force, more nitrogen pushing onto the biochar surface. But eventually it levels off. Either all the available absorption sites get saturated, they're full up,
Okay.
or perhaps the pores get blocked, limiting further access.
Makes sense. What about temperature?
With temperature, it's a bit more nuanced. Increasing it, say, from room temperature like 25 degrees C up to maybe 35 degrees C generally boosts absorption.
Why?
Because the molecules just have more kinetic energy. Everything's moving faster, diffusion is quicker, the absorption process speeds up.
It's always a but.
Well, yeah. There's a delicate balance. If you go to much higher temperatures, especially if the pH is also high, the ammonia nitrogen can actually start to volatilize, turn into ammonia gas, and just escape the water.
Ah, so it doesn't even get a chance to absorb.
Exactly. So too hot can actually limit the overall absorption.
Tricky balance. What about pH? That's always critical in water chemistry.
Absolutely vital. The pH of the solution creates this kind of Goldilocks zone for absorption, typically best between pH 6 and 9.
Why that range?
If it's too acidic, so low pH, you have lots of hydrogen ions floating around, and they basically compete with the positively charged ammonia ions for those binding sites on the biochar.
Okay, competition.
Yeah. And if it's too alkaline, say above pH 9, the ammonia nitrogen starts to convert into its unionized form. Ammonia is dissolved in water, which isn't as easily absorbed by the mechanisms biochar typically uses.
So pH 6 to 9 is the sweet spot.
Generally, yes. Now, looking at the biochar itself, carbon content, see, it has a significant impact, but it's also a bit of a balancing act.
How so?
When carbon content is lower, say less than 60%, increasing it often leads to better absorption. This is likely because higher carbonization often creates more surface area.
Okay.
But interestingly, when carbon content gets really high, say above 75%, absorption performance can actually start to decline.
Why would that happen?
The thinking is that very high carbonization can sometimes reduce the number of important surface functional groups, especially oxygen-containing ones, which play a key role in chemically binding the ammonia.
Ah, okay, so it's not just about surface area.
Exactly. And speaking of those functional groups, the OC ratio, oxygen to carbon, was highlighted as crucial. It's a direct indicator of those oxygen-containing groups, which facilitate strong electrostatic interactions with the positive ammonia ions.
Got it.
Similarly, the HC ratio, hydrogen to carbon, matters. Higher HC ratios often suggest more hydrophilic or water-loving groups on the surface, which can enhance absorption through things like hydrogen bonding and other electrostatic interactions.
Okay. And what about ash content and the physical structure, the pores?
Right, ash content. It showed a fascinating nonlinear effect. Sometimes minerals in the ash can actually help by influencing the surface alkalinity in a positive way.
Oh, interesting.
But if you have too much ash, it can physically block the micropores, basically reducing the number of effective sites where the nitrogen can actually stick.
Another balancing act.
Seems to be a theme. And finally, the pore structure, total pore volume, V, and specific surface area, BET. Of course, they're important. You need pathways for the nitrogen to get to the adsorption sites, and you need surface area for it to stick.
Right.
But what this study really underscores is that physical adsorption isn't the only thing going on. The chemical interactions, electrostatic attraction, ion exchange, surface complexation involving those functional groups are equally, if not more, complex and absolutely critical to the overall process. It's a combination of physical and chemical factors.
Okay, that makes a lot of sense. So what does this all mean for practical use? The researchers didn't just stop at finding the best model, they made it accessible, right? Which is fantastic for a real-world application.
Absolutely. And this is, I think, a really cool part. To help get this knowledge out there and used, they developed a Python-based graphical user interface, a GUI.
So basically a user-friendly app,
Yeah.
essentially, yeah. A simple piece of software. You can input 10 key parameters, things about your biochar, the plant experimental conditions like temperature, pH, initial concentration, click predict, and it instantly gives you the estimated ammonia nitrogen adsorption capacity based on that powerful cat boost model.
Wow.
This tool can genuinely help optimize experimental design. You can virtually test different scenarios before you step into the lab, potentially saving a huge amount of time and resources. It speeds up decision-making for environmental engineers.
That's incredibly powerful for anyone working in water treatment or material science in this area. What are the broader impacts or advantages of this study beyond just the app itself?
Well, I think this research offers several key advantages. One big one is transparency and reproducibility. They made all the Python code, the raw data they compiled, and even the GUI application code openly available on GitHub.
That's great.
Yeah, it allows other researchers to verify the findings, build upon them, maybe adapt the model. It really accelerates progress when science is open like that.
Also, because they used that significantly larger data set and looked at a comprehensive set of features, the resulting model is likely more robust and generalizable than previous attempts.
Yeah.
It deepens our fundamental understanding of how biochar actually works in this context.
Right.
And, critically, as we've said, this whole methodology facilitates efficient adsorbent selection and process optimization.
Yeah.
It drastically cuts down the need for purely trial and error, labor-intensive, costly experiments.
Saves time, saves money, gets to solutions faster. Now, every study has its boundaries, right? What are the limitations the researchers acknowledged? What can't this model do yet?
Of course, yeah. Good science acknowledges its limits. They note that the data, while extensive, doesn't cover every single possible condition or biochar type you might encounter in the real world.
Okay.
And importantly, its current focus is on ammonia nitrogen removal in relatively controlled settings. It doesn't yet extend to predicting performance in complex, real wastewater scenarios.
Where you have lots of other stuff mixed in.
Exactly. Multiple contaminants interacting, fluctuating conditions. That's a whole other level of complexity that future work needs to tackle. Also, as we discussed, the deep learning models didn't perform as well, suggesting maybe they need further development or different approaches to be effective for these kinds of specific structured data sets.
Okay, so what's next then? Where does this kind of research go from here? What are the future directions that excite you?
Well, the path forward definitely involves tackling those real-world complexities, incorporating the effects of multiple contaminants, understanding how these models perform with actual industrial or municipal wastewater.
Making it more applicable?
Exactly. And continuously enhancing the models themselves, making them even more accurate, and perhaps finding ways to make deep learning more effective, even with these more specialized, maybe smaller data sets. It's a really dynamic field, integrating material science, environmental engineering, and data science. Lots of potential still.
Wow. From the silent threat of nitrogen pollution to the natural filtering power of biochar, and then harnessing the predictive capabilities of machine learning. What a journey we've taken today.
It really is incredible.
It's amazing to see how advanced computation is helping us solve these really critical environmental challenges much more efficiently.
Yeah, this deep dive truly demonstrates the power of integrating diverse scientific fields. It's not just about predicting an outcome, right? It's about gaining a deeper understanding of that intricate dance of variables. And then using that understanding to engineer better solutions to build a cleaner, more sustainable future.
So here's a final thought for you, our listener. Consider this. If machine learning can so effectively optimize something as complex as biochar adsorption for cleaner water, revealing these key factors and relationships we might have missed just doing lab work, what other environmental puzzles could it unlock? What other complex natural or engineered systems could benefit from this kind of intelligent analysis where maybe we haven't even begun to connect the dots yet? How might this kind of interdisciplinary thinking, combining nature-based solutions with data science, apply to problems you encounter? Something to think about. We'll be here next time for another deep dive.